Hello everyone, I am Rekha Mudappathi, a postdoctoral researcher at Arizona State University. Today I'll present MPET, a computational framework for studying how intracellular transport regulates surface protein expression and contributes to disease. Intracellular protein transport or ICT is a process that delivers proteins to the cell surface where they perform essential functions such as antigen presentation and immune signaling. This process is coordinated by a group of genes called ICT or intracellular transport genes. When ICT is dysregulated, surface protein expression can change even when mRNA levels remain unchanged, contributing to diseases such as cancer, infections, and neurodegenerative disorders. However, most current analyses directly relate mRNA to protein abundance and overlook this transport layer. To address this gap, we developed MPET, a computational framework that integrates site-seq data to quantify how ICT regulates surface protein expression and contributes to disease. This slide provides an overview of the MPET framework. MPET stands for Modeling Protein Expression and Transport. Starting with site-seq data, which simultaneously measures gene expression and surface proteins, MPET analyzes how ICT contributes to surface protein regulation. Module 1 identifies ICT genes associated with surface protein abundance after accounting for the expression of the corresponding coding gene. Modules 2 and 3 then use mediation analysis to determine whether these transport effects contribute to disease either through individual ICT genes or through the combined effects of multiple ICT genes. Finally, Module 4 integrates these relationships into regulatory networks, providing a systems level view of how ICT influences surface protein expression and disease phenotypes. MPET is built around what we call putative transportation trios or PTTs. Each trio consists of a surface protein, its coding gene, and one ICT gene. For every combination, MPET tests whether the ICT gene is significantly associated with surface protein abundance after accounting for the expression of the coding gene. Significant associations are retained as PTTs and serve as the input for the downstream analysis. To demonstrate MPET, we applied it to a publicly available site-seq dataset from COVID-19 patients. The dataset included samples from 12 individuals comprising approximately 60K cells. We focused our analysis on CD16 plus monocytes because this population exhibits marked immune dysregulation in severe COVID-19, making it an ideal model for studying how ICT influences surface protein expression. We evaluated over 61K ICT trials in CD60 plus monocytes. Across all groups, we identified more than 11K PTTs. We observed that highly expressed surface proteins recruited large number of ICT genes, suggesting that proteins with greater surface abundance require more extensive ICT machinery. To evaluate the contribution of intracellular transport, we compared models with and without ICT genes. Including ICT genes consistently improved association between coding gene expression and surface protein abundance, demonstrating that ICT provides additional information beyond gene transcription alone. Here we examine the overall ICT regulatory patterns. The heat map shows many-to-many relationship where multiple ICT genes regulate same surface protein and individual ICT genes influence multiple proteins. When we compared these patterns with conventional mRNA protein relationship, we observe very different clustering. This indicates that intracellular transport represents a distinct layer of post-transcriptional regulation beyond gene transcription. We next examine cases where surface protein expression changed without corresponding changes in mRNA. These protein-mRNA mismatches suggest regulation beyond transcription, which is precisely what MPET is designed to capture. CD69 provides a clear example. Although CD69 surface protein was significantly increased in severe COVID-19, its mRNA expression remained unchanged. Using MPET, we identified disease-associated ICT genes that mediate this increase in surface expression, demonstrating that enhanced ICT, not transcription, is responsible for the observed protein level changes. We next applied MPET to CD69 using mediation analysis. We identified 138 DAICTs. These are ICT genes whose effects on disease are mediated through the surface protein. Most of these DAICTs showed positive mediation effects, indicating enhanced transport of CD69. These findings demonstrate that increased CD69 is driven by transport activity rather than changes in transcription, illustrating how MPET uncovers post-transcriptional mechanisms underlying protein regulation. We next examine HLA-DR, a key antigen presentation molecule and an established marker of immune dysfunction in severe COVID-19. As expected, HLA-DR surface expression was reduced by approximately 25% in severe cases compared with healthy controls and mild disease. We then use MPET to investigate whether this reduction could be explained by dysregulated ICT. This network summarizes the regulatory relationships identified by MPET. HLA-DR is the central mediator connecting ICT genes to disease severity. ICT genes shown in green act through HLA-DR. Those in yellow influence disease both through HLA-DR and directly, while blue genes have direct effects independent of HLA-DR. Together, this network provides a systems level view of how ICT contributes to disease. To better understand the biological functions of these ICT genes, we performed pathway enrichment analysis. More than half of the mediated ICT genes were associated with endocytosis, suggesting that enhanced endocytosis leads to intracellular retention of HLA-DR rather than reduced transcription. Several key ICT genes further supported this mechanism, indicating that dysregulated transport underlies the loss of HLA-DR surface protein expression in severe COVID-19. In summary, MPET is a computational framework that models how intracellular transport regulates surface protein expression at single cell level. By integrating transcriptomic and proteomic data, MPET explains mRNA protein discordance and uncovers transport mediated regulatory mechanisms underlying disease. Together, these findings establish intracellular transport as an important layer of post-transcriptional regulation and provide a new framework for studying surface protein regulation using the single-cell multiomics. Thank you.